this.
Hi everyone, welcome to the AWM Pitt Seminar. Today we have two speakers. Our first speaker is Mariya Savinov. She is a PhD student from New York University. And our second speaker, Anna Thomas. She is a PhD student from University of Pittsburgh. So Mariya will talk about modeling the cytoskeleton, how friction can guide its dynamics. Mariya, you can go ahead.
All right, great. Yeah, this is a relatively small group, so feel free at any time to interrupt if you want to ask a question or confuse, because I don't think everyone here has a background in like math bio or cell bio especially. But yeah, thank you for the introduction. Thank you so much for having me speak with you guys today about how my work on modeling the cytoskeleton and how friction can guide its dynamics. But just to start, a little bit about me. As I said, my name is Mariya. I actually graduated from Pitt in 2020 with a BPhil in math and physics. I worked with Bard, who's here. And my background with Pitt goes far beyond that. I actually grew up in Pittsburgh, and my parents are faculty at Pitt, so I'm pretty sure they're both in the Zoom right now, so hello. But yeah, I'm currently fourth year math PhD candidate at NYU at the Courant Institute of Mathematical Sciences. I'm in this very exciting building with big windows, which is a nice change. And I work with Alex McGillner on cell biology problems. So I'm particularly interested in mechanical phenomena in cellular biology. Beyond that, I care a lot about like collaborative research. All the work I've done has been in collaboration with experimentalists. I care about DEI in academia, and I have hobbies like board games and video games. So just to personalize me beyond just my work. But yeah, so a little bit of a crash course, a little bit in some like the cytoskeleton / cell biology. There are lots of different cellular behaviors and processes that matter a lot for cell life and as such like organism life. Two of the big things that matter for cell life are motility and division, for example. So on the left here, this is a type of wound healing cell in fish skin. So I think the way they usually get these on glass slides is they literally just scrape the scales off the fish, and then these just come off. And they're a very classic example, like they're a very classic experimental system because they move really fast. And they have very persistent properties that we can analyze into how they move. So they're a classic example of motility. On the right, this is a GIF of like a cell division process in the sea urchin embryo. So we can see we go from 4 cells to 8 cells on the edge. And both of these things, both of these processes are dependent on the dynamics of the cytoskeleton. So when I say the cytoskeleton, right, you think cell skeleton. I quite literally, it kind of feels like a skeleton, right? If you take a look under the hood, right, this cell I think is like 10 microns in size or so, or not 10 microns, maybe tens of microns in size. And if you look at like under the hood, sort of what's happening here, and you zoom in on a 1 micron by 1 micron square, which is what we see on the far right here, you'll see that actually it's composed of this really, really dense interlinking filament network. These are like biofilaments made of a really, really abundant protein in eukaryotic cells called actin. So actin, if we take it alone, right, actin is a protein that self-assembles into these filaments. These filaments tend to be about 7 to 9 nanometers in length, but when you add other things to it, they tend to either grow longer or assemble into higher order structures. These higher order structures tend to behave viscoelastically, which I will get back to later. In general, in the cell, you don't just have you don't just have actin and its assembly, you also have a bunch of proteins that will interact with it. So you could have, for example, motor proteins, which there are a lot of like classic examples of motor proteins, classic like little videos, toy videos of them walking along the filament. And as they walk, the filament slides in the other direction. There are, of course, like cross-linking proteins, branching proteins, and essentially all they do is they modify the way the filaments grow and the way that they interact. But my favorite protein is myosin. Myosin, in the presence of antiparallel filaments, so these filaments, they're polar. They actually have one end that grows faster and one end that grows shorter, so they have direction. And the motors tend to walk in a specific direction along the filament. So if you take 2 antiparallel filaments, the motor is walking this way along one and this way along the other. So it causes initially a contraction process and then an extension process. But due to an abundance of factors and some nonlinearities, if you take like this big soup of actin and this big cross-linked interlinking network of filaments, generally when you add these motors, they're consuming energy. When you add these motors, usually you primarily see contraction. And we can't really get into why and how, but when we think back to the sea urchin embryo example, you can see that just before we see this division into 8 cells, you're seeing this bright patch. This is actin is accumulating. And in fact, it's accumulating in a ring along the edge. And the motors are causing all of this ring to get smaller and smaller and smaller to divide it into 8 cells. Well, I guess it doesn't go all the way, but for the most part, this is one of those examples of how adding this activity to actin is resulting in the behavior that we observed in the cell. So going back to the example of the moving cell, right? We know that there's a big, really dense actin network, primarily aligned toward the front in the cell. We conceptually and experimentally know that in the presence of these motors, there's contraction. So if you actually look at where the motors are in these motile cells, the myosin motors are primarily toward the rear. So we can sort of think of it this way, right? The actin, as it grows, it's growing and pushing forward in the front of the cell, but the myosin motors are making sure that the rear doesn't get stuck, right? It's contracting the rear and pulling it forward so that the cell can actually move forward in the end. Now that's one explanation, of course, but when it comes to actin-myosin dynamics in general, they're a balance of many, many things, right? Of course, you have these contractile myosin forces, and it matters where they are in principle. But you can also think of this whole actin network as like a material, right? And when you deform a material, there are forces because of that. There's also, what I haven't mentioned at all, is there's also friction and then adhesion, right? Not only is the cell pushing forward in the front and retracting in the rear, it's actually also sticking adhesions as it goes forward. And finally, there's also the aspect of geometry, right? In this particular case of this kind of cell, there's a lot of research done as to how certain, like why it has this particular shape and how this shape or variations of it aid its kind of motility, right? So this is what Alex and I sort of wanted to do, is we wanted to look more into what are the relative importance of all of these 4 things, right? Does it matter the most, right? What matters the most in some sense? You could ask this question as to, well, could you have a small variation in items 1 and 2, but ultimately, if you have the same geometry, do you have the same behavior, right? So what we've done is that our collaborators at the Cytomorpho Lab, which is located in Paris and in Grenoble, they had this sort of experiment, which is, let's consider something which is in vitro, meaning we're going to consider it in the lab, not in the cell, and we're going to control what we put into it, right? What they can do is they can choose glass surfaces of chosen shape or size. So you can consider squares, rectangles, discs, and these glass surfaces, they're about like tens of microns. So like, think of like a really big cell, basically. You can coat these surfaces with some mix, and you can isolate them by essentially putting a special kind of polymer on the outside so that nothing else can interact with that polymer. You can add an assembly promoting protein to make sure that when you grow the network, it only grows in this square. And then finally, we want to add actin, right? So in this experiment, right, all you've essentially done is you've said, okay, actin, you should only grow in this domain. And then you put in basically just like monomers of actin with a couple other proteins and see what happens. So very quickly, in about 10 minutes, I kind of want to say, we see this assembly of a really dense branched actin network, just like we were seeing in that electron micrograph of the fish cell. So I love watching this video personally, because you don't always get this really high resolution growth of actin, and you can actually see their diffusion a little bit. But regardless, this happens very quickly, right? In like within 10 minutes. So the question becomes, okay, what if we add activity? What if we add motors that consume energy and result in like active forces? What happens in that case, right? Because that's the case that actually matters when it comes, rather, it's one of the cases that matters in these biological processes that we're discussing. So in this video, I'm going to try my best to explain it carefully. So on the left side, we have actin in red, and on the right, we have myosin in green. So I want you to focus on just the actin side. So when you see like bright red, it'll kind of clear up very quickly. That essentially means that there's a high density of actin. Okay. So as the movie plays, we see that in this short period of time at the beginning, we see the assembly of this network throughout the square. right? But after the network is assembled, right, as the network is assembling, we also see the aggregation of myosin, right? So you see these bright green spots. They sort of appear right from the beginning almost, and then they kind of get stronger and brighter and bigger up to a certain point. So these are actually really dense aggregates of myosin that we didn't control where they appeared. They just appeared randomly, 3 of them. And once enough time has passed and the whole network is assembled, it all contracts to the center of the square. So I can play this again. We see that at the beginning, the network assembles in the domain of your choice. But because you have these motor proteins, which are walking along the filaments and causing contraction, they contract the whole network, but importantly, they don't contract the network to themselves. They actually also move, and the whole network contracts to the center of your shape, to the center of the square. It's really hard to see on the left side because there's so much actin that it's really hard to distinguish any structure here. But the fact that all the myosin is in the center really tells us the whole network really compacts, at least centrally, symmetrically in the center of the square. And we can do a bunch of different experiments with this. And here in different colors in like light blue, dark blue, red, and green, we have a couple different experiments with where in these experiments, myosin randomly accumulated into these different places. And if you follow the trajectories of these bright spots, just like you can put a little dot and you follow that dot, right? We see that the trajectories all go to the center of the square. And in general, in the like many experiments that our collaborators have done, we always see contraction of these networks to the center of the square. So really, remarkably, even though myosin is producing local forces, it's resulting in this global contraction that doesn't really depend on its initial distribution, right? The final point of this contraction is insensitive to myosin distribution, which to me was really initially very surprising, right? Because you think, okay, well, the motors are the ones who are causing the activity, right? Shouldn't they have some say in where they go? But it seems to be that the geometry matters much more here, right? So I guess what are the components that matter, right? We've tried a couple of different geometries, and it's always the geometric center to which the network contracts to. So one thing that we can change to sort of figure out, okay, well, if myosin isn't really the governing, I guess myosin distribution isn't the governing parameter in this behavior, what is? So one thing that they can do in the lab, which I think this was like years of work, basically, is that they were able to micropattern such that half of the square is like a lipid bilayer, which is essentially kind of like a membrane-like material. And on the right side, you have glass. So the lipid side is going to be lower friction than the glass side because the lipids can diffuse freely at a slower rate, but they can diffuse. So in this case, just like before, we see the assembly of the network in the square and the aggregation of myosin, these motors, into these tight aggregates. But this time, when contraction happens, it goes to the right side of the square. So let it play one more time so you guys can see. So we can do lots of experiments just like before. And in the different experiments, maybe you have one myosin aggregate that's only on the lipid side. And yet somehow it almost, to me, visually, it looks almost like it directly goes to this contraction point on the right side. And notice there's symmetry vertically, but the asymmetry is only where you have asymmetry of friction. So the experiments suggest that the contraction point is determined by friction in a way that, once again, is insensitive to the myosin distribution. We just need myosin to cause contraction, but it doesn't matter where it is. So, of course, I didn't title this talk how we can do experiments, right? This is a modeling problem, right? Just to summarize, right, in the experiments, we're seeing that in the homogeneous case where everything is the same surface, we see contraction to the geometric center. But when the surface friction changes, we see contraction to the higher friction side. So to sort of make sure that we understand what really are the components driving this behavior, we turn to modeling and simulation. First, right off the bat, right, when you're doing modeling, you want to simplify in some way or make some assumptions, which in this case, what we see is roughly 2D dynamics. So we're going to use a 2D model. We need to pick a rheology for the network, right? We need to have it that it responds to internal deformations in some way. So in principle, we have a couple choices. It could be like an elastic solid where stress is proportional to deformation. So that's sort of like a spring, right? The stress of that spring is exactly proportional to how much you stretch it, or rather roughly proportional to how much you stretch it, at least in the linear case. You could have something which is more like a fluid, but is very viscous. So the stress is proportional to the rate of deformation. Classic example of a viscous fluid, as some sort of intuition, would be honey, right? It matters more how quickly you pour it, as opposed to like whether you're pouring it at all. But in general, we could consider something that's viscoelastic too, right? I found this GIF online, and I thought it was somewhat representative of this idea of viscoelasticity, where this piece of Jell-O is getting hit with a bowling ball really, really quickly. And initially, it almost feels kind of like, at some point, it kind of almost looks like a fluid to me, like kind of like water. But in general, overall, it maintains its shape, right? In the long term, for the most part, it looks like to me anyway. So there are two different ways, two very classic examples of like linear viscoelasticity would be Kelvin-Boy models and Maxwell models. But in general, lots of people have done experimental work on actin networks and on the cytoskeleton. And it's generally been observed that these networks are viscoelastic in the way where on short time scales, they're elastic, but on long time scales, they're viscous. So to give you guys a bit of intuition as to why we would expect that, think of it this way, right? You have this, I wish I had brought the picture back at this point, but you have this dense network of filaments, right? On really short time scales, they're all interlinked, they're branching, they're connected. It's kind of like a solid. So if you try to stretch it, it doesn't want to stretch, right? Or if you try to compress it, it's not necessarily going to want to compress because this filament is attached to that filament, and that causes some sort of pressure and tension on everything. However, if you wait long enough, right, these filaments, they're assembling, they're disassembling, all of these different proteins, they're attaching and detaching, there's diffusion, right? All these different aspects mean that over long times, everything rearranges in the end, right? So it's more fluid-like in the end than it is solid-like. So we want to consider, for this particular experimental problem, we want to consider a viscoelastic model, which is elastic on short time scales and viscous on long. So if we think of it as like a viscoelastic material, let's, at least to simulate it, let's break it up into pieces, right? Let's break it up into, let's coarse grain it so that each of these green nodes represents a patch of the network. And each of these patches of the network are connected to their neighbors with a certain choice of rheology, right? When it comes to the actual motors and their effect on the network, what we're going to have is that we're going to have that we choose where the aggregates are located by choosing a center, and then have there be sort of like a radius of effect at which this center of this aggregate of myosin generates some tension. So to walk us through what the governing equations are, each of these links between these patches of network is essentially, at least in short time scales, it's basically a linear spring with potentially some additional tension. So this part on the right is a spring force for if you squish these two nodes together or stretch them out, you'll have some sort of linear spring force. And then potentially, if you pick like a part of the network that's close to this myosin spot, there's also some sort of active stress, which is causing it to shorten, causing it to contract. Now I say linear, but it's not really quite linear. We actually want for this network to be harder to stretch than it is to compress, right? We want something which is a quote unquote a cable network. So a really classic example of this, or rather a nice example of this would be if you think of a fish net that has a lot of holes in it, right? It's hard to stretch it out from all directions, but it's really easy to crumple up, right? So we're not going to consider something so dramatic, but in general, we will consider something that is harder to stretch than it is to compress, because these filaments can buckle, but they're hard. If you stretch them, they don't want to be stretched. So now the viscous part comes into the fact that this rest length, Lij, is the rest length between node I and node J. We want it to relax over time, which means that over time, with some time scale tau, we want it to assume a new reference configuration. So essentially what we want, because these networks are turning over and there's assembly and disassembly, we want that if you, for example, if you squish it and you hold it, right, into a new shape, we want that new shape eventually, if you hold it for long enough, to be its relaxed configuration, because the filaments have moved, things have detached, things have reattached. So now it doesn't care that initially it was a square. If I hold it tight into a circle, eventually it'll say, okay, circle is fine with me, right? So this is where the viscous part comes in, where at long time scales, it's going to relax and be more fluid-like. And finally, we need something to balance out these forces. So we're going to have drag with the surface. which is just a very classic sort of generic drag term of some drag coefficient depending on where you are, times the rate of change of the position. And then the DAI is like an area weight. So that way, if you coarse grain it so that all of the nodes are, there's more nodes, then everything scales accordingly based on how many nodes you have. So yeah, this is the drag term. So overall, these are all of our governing equations for our model. These are sufficient. You'll see that this is in fact sufficient to describe the behavior that we see in the experiments. Now, skipping right to the simulations, right? No one gets to see the months of coding work that go into these sorts of things. You just get to see the pretty videos. So we're going to spend some time on the pretty videos. Now, on the right is the case where the whole surface is the same. But on the left, we have the case where in red, which is the left half of it, it's a lipid, and on the right half, it's glass. So I'm just going to play the homogeneous case first. So these bright yellow dots are essentially where we choose to put the myosin aggregates. And what they're going to do is they're going to contract the network around it, eventually contracting the whole thing to the center of the square, which is exactly sort of what we see in experiments, though you might argue with me and say, well, you picked something super symmetric, so of course it's going to like symmetrically converge to the center. So bear with me one more slide and you'll see that in fact it doesn't matter. Meanwhile, you can consider the heterogeneous case where it's half lipid, half glass. And in that case, the network converges to the right side of the domain, just like in the experiments where it's symmetric vertically, but asymmetric horizontally. So now those of you who don't believe me, let's consider just one myosin aggregate that's in the bottom left corner, right? This is a very asymmetric case. And we'll see that, in fact, that one myosin aggregate contracts the whole network to the center in the homogeneous case, but to the glass side about like 7 microns far in the heterogeneous case. And just like in the experiments, we can do lots of simulations. I like to call them experimental simulations sometimes. So different colors here represent different experimental cases. Different simulation cases, which actually do try to match the experimental positions. And in very robustly, in almost all cases, we see convergence to the center when you have a surface which is all the same friction, and convergence to the higher friction side when you have low friction, high friction crossover. And we also plot the experimental points here. So we are slightly underestimating the experimental result, which I do have an explanation for, but I don't think we have time, so happy to answer that at any point in time. But we don't just reproduce the guiding rule of friction, right? We do a couple other things. Or I guess rather we qualitatively capture some of the other, we capture other qualitative results from the experiments. For example, we see, just like in experiments, that corners are rounded by contraction. So if you consider this example case of the 4 aggregates, very quickly these corners of the square collapse, essentially, and the whole thing is rounded, which is what we see in the experiments. We also see that the interior doesn't deform until later stages, because notice how the interior here on the right side at 9.86 minutes hasn't really changed much from the initial time, which we also see in the experiments. You can photo bleach a grid pattern, and that grid is barely deformed in the interior as opposed to the exterior. So that's essentially most of what I have for you. There's just so much more work I could talk about, so much more I could talk about with this, but I wanted to give you guys the main points of how we can model the cytoskeleton and how friction can guide the dynamics and be the governing factor in determining them. If you are interested in learning more about this, this work is published in PNAS. It was really fantastic to work with this experimental group. And of course, thank you to my advisor, Alex, for all his help with this. With that, I'll leave you with my email and my Twitter handle for anyone who uses academic Twitter. It's very common and popular in biology, so I've adopted it. But yeah, thanks again for having me, and I'm happy to answer any questions if we still have some time.
Thank you so much for the talk. If anyone have any question, you can ask.